Hello, and welcome to Part 3 of Chapter 10, where I will discuss Section 10.6, Cell Cycle Control. This section focuses on multicellular organisms. In multicellular organisms, cell cycle is controlled by external signals, known as growth factors, and internal signals, known as cyclins and CDKs. Growth factors are typically hormones that are released by the organism that come in contact with the cells and that they're signaling. Cyclins and CDKs cyclins are proteins, and CDKs are also proteins, but they're enzymes, they're kinases. An example of how they work: when growing cells in a petri dish or in a culture, the cells will only grow one layer thick and then they'll stop growing. It's because when the cells come in contact with one another, membrane receptor proteins will activate a signaling pathway into the cell that stops the action or inhibits CDK. So a single layer of cells will not grow into multiple layers in a culture plate. Now, growth factors work by triggering cell division, so they promote cell division. And growth factors will be released by blood clots. That's a good example. So if you cut yourself or get a bruise where you've damaged cells and tissues, the clot will form to stop the bleeding, but then they'll also release a growth factor so that the nearby cells will be triggered to go through cell division. Along with these signals, there are also checkpoints for the cell cycle to keep control of everything. So let's go back to our cell cycle figure that was introduced in Part 1 of Chapter 10. And this time we've introduced the checkpoints into the figure. So the first checkpoint is the G1s checkpoint up here in the top left corner. And this checkpoint checks the size and health of the cell. So when a cell gets a signal to divide, it goes into the G1s checkpoint, checks to make sure it's of mature size, it's healthy, but most importantly, it checks the integrity of the DNA to make sure that the DNA is intact and is in a good shape to be copied correctly. If everything looks good, the appropriate cyclins will be released and the cell will enter the S stage, makes a copy of its DNA, then goes into G2, where it encounters the second checkpoint, the G2/M checkpoint. This checkpoint checks that the DNA was replicated completely, correctly. So it's checking the integrity of the DNA again, but this time it's looking at both copies to make sure both copies are okay. If everything is okay, the appropriate cyclins will be released, and then the cell will enter mitosis. The third checkpoint happens in metaphase, when the chromosomes line up at the metaphase plate. This checkpoint ensures that all the chromosomes are attached to the spindle and that they're at the metaphase plate. If everything looks good, the appropriate cyclin is released, and then the cell will go through anaphase and finish out mitosis and possibly cytokinesis. Now, at any of these checkpoints, if things don't look good, if there's mistakes seen in the DNA or the copy of the DNA, or there's chromosomes not lined up properly, then the cell will try and correct the situation, fix the problem. If it can, then it will pass the checkpoint, the appropriate cyclin released, and it will go on. If it cannot fix it, then the cell will hit the auto-destruct button known as apoptosis. The term apoptosis is not introduced in your book until Chapter 16, which we're not covering, so I'll just refer to it as programmed cell death or hitting the auto-destruct button. Now, it turns out that the checkpoints are critical for preventing cancer development. Cancer is a multi-stage process. It involves the disruption of normal cell division and behavior, and it's initiated by mutations that may occur in genes known as proto-oncogenes or tumor suppressor genes. In most cancers, mutations are found either in one or both of these genes. So we will talk about how mutations in either of these genes affects the cell cycle and promotes the development of cancer. Let's start with the proto-oncogenes. It's kind of a long, complex name. Okay, so this is an illustration that shows you how proto-oncogenes work. These genes encode proteins that promote the cell cycle. The analogy given is like it's the gas pedal of a car. When you want to divide, you want to go, you push down the gas. There's a proto-oncogene that codes for a protein called the Ras protein, this one here. Okay. It is a membrane protein. It's present in the plasma membrane of a cell, and the way it works is it's part of a signaling pathway. So when a growth factor is detected by a cell, which tells the cell that it needs to divide, the growth factor binds to the receptor, and the receptor sends a signal to the Ras protein, which then initiates a signaling pathway to the nucleus, which turns on certain genes that make proteins that begin the process for cell division. When a proto-oncogene is mutated, it becomes an oncogene. You just drop the proto part. And oncogenes basically are build mutated proteins and act as if the gas pedal is always pressed down. It's stuck on. Let's show you an example of what happens if this Ras proto-oncogene gets mutated and becomes an oncogene. So in this second figure, we have a mutated Ras protein because it was made by an oncogene. And it is sending a signal to the nucleus to go ahead and make the proteins for cell division, even though there is no growth factor present. So the gas pedal's stuck on, even though the cell's not being told to go, not being told to divide, it's going to divide anyway. And it's going to ignore those checkpoints. It's just going to barrel on through, even if there's mistakes in the DNA, even if the chromosomes are not lined up or attached to the spindle. It's just going to keep going, keep going, keep going. So the chance of more mistakes occurring increases almost exponentially, which leads to many, many cells rapidly dividing and not carrying on the normal function they're supposed to be doing, and those are cancer cells. So let's look at the second type of mutation that's found in many, many, many types of cancer, and that is tumor suppressor genes. So tumor suppressor genes encode proteins that actually stop the cell cycle. They act as the brakes of the car. If a mutation occurs in one of these genes, then the brakes don't work. It's like cutting the brake lines, and even though the cell's trying to stop, it can't. The example your book uses is a well-known tumor suppressor gene known as the P53 gene. It halts cell division if damaged DNA is detected. So in this top picture, you will see a normal P53 protein made by the P53 gene. When it detects damaged DNA, right here, it will then initiate repairs. A DNA enzyme will come in and repair it, and then it will allow the DNA to be copied and cell division to continue. If the damage is not repairable, then it will hit the auto-destruct button and initiate the programmed cell death. If this tumor suppressor gene is mutated, it cannot make the normal P53 protein, and so the damaged DNA does not get fixed, and it will continue into each generation of cells, and the checkpoints are ignored, and you end up with cancer. There are different stages of cancer. Most of you know or have heard of some of the terms before: benign, malignant, metastasis. Benign cancer is cancer that is not invading surrounding tissues or affecting organs. Malignant cancer is cancer in which it is interfering with the normal functioning of the surrounding tissues or organs and is considered lethal. And metastasis is cancer that's grown so much and gone through so many mutations that it's developed the ability to spread, to move to other unrelated parts of the body. So, sorry to end on such a down note, but this is the end of part 3 and chapter 10. Thanks for watching.